Traditional positional cloning involves linkage mapping to identify the chromosomal location of a mutation producing a trait, followed by candidate sequencing of genes in the vicinity of the linked markers to identify which gene has the causal mutation. In the last few years, an alternative approach to mutation identification has been applied. This process, high-throughput sequencing, essentially sequences all of the genes and then uses computational approaches to filter the sequence data and identify the causal mutation. This session will describe and discuss that process to achieve the learning objectives outlined here. The concept that mutations can be identified by sequencing all of the genes is a benefit of knowing the genome sequences of humans and now many other organisms. Humans were among the first complex organisms whose genome was sequenced due to investments in the Human Genome Project. The project to sequence all 3.2 billion bases of the human haploid genome was completed in 2000 after about 10 years of work and cost about $1 billion. Along the way, lots of new enabling technologies were developed that continue to benefit the genetics community and the larger research community as well. This reference sequence is not the sequence of a single person, but rather an amalgamation of the sequences of a number of people. Once we had a reference sequence for the human genome, which can be looked up and searched online, all subsequent genomes were just resequencing efforts that used the first genome as a guide. Consequently, the second genome took only a year and $30 million to complete, and the third 6 months and about $3 million. Things progressed rapidly from there, with the fourth and fifth genomes completed more quickly and for a lot less money. The sixth and seventh more efficiently still, and the eighth genome even faster and cheaper. It is now not possible to keep track of the number of human genomes that have been sequenced, but the time and cost has continued to come down so that under some circumstances, a genome can be sequenced in about a day for about $1,000. Furthermore, similar benefits have been achieved for all of the model organisms we study and many other species too. The main innovation that enabled this progress is the dramatic cost reduction afforded by high-throughput sequencing. Technological innovations allowed the costs of sequencing to plummet as we went from sequencing each molecule on its own by Sanger sequencing to a process called high-throughput or next-generation sequencing, in which all of the sequences in a genome are determined simultaneously. Here, I will describe and contrast the conventional Sanger sequencing analysis with high-throughput sequencing. In the conventional approach, DNA fragments are sequenced one by one. The DNA is fragmented in some way, and each of the resulting pieces is cloned in bacteria. The bacteria are propagated and the DNA isolated so there is enough to sequence. Sanger or cycle sequencing is then used to determine the sequence. Sanger sequencing involves enzymatically extending a primer that is complementary to a template and upstream of the DNA to be sequenced. DNA polymerase extends the primer, and the mixture contains the enzyme plus a mixture of deoxynucleotide triphosphates, or DNTPs, spiked with fluorescently labeled chain-terminating dideoxynucleotide triphosphates. The ratio of dideoxy to deoxynucleotides determines the probability of incorporating each type of nucleotide. When a dideoxynucleotide is incorporated, synthesis is terminated, and the partially elongated chain is labeled with the fluorescent tag associated with a particular dideoxynucleotide. In this example, dideoxy-C has a yellow label, DDT has a blue label, DDA has a red label, and DDG has a green label. For the molecules that incorporate a deoxynucleotide, they can incorporate the next base, which can also be terminated if a labeled ddNTP is incorporated in the next step. The second base has a label corresponding to its identity and is one base longer than chains terminated in the previous step. Thus, at each step, there will be some molecules terminated and labeled, and this process can go on for hundreds of nucleotides. The collection of labeled fragments, all differing in size by one base, are separated by gel electrophoresis. Then, by reading the fluorescence signal, the sequence can be determined by the color at each position. In the example, the sequence is yellow, blue, red, blue, green, yellow, blue, yellow, green, corresponding to the sequence CT-AT, GC-TCG. This is the way most of the sequences in the Human Genome Project were determined, with each sequence aligned with its neighbor by the overlapping sequence they shared. High-throughput sequencing uses very similar chemistry, with the major difference that all of the fragments are sequenced at the same time. Once fragmented, adapters are attached to the ends of each fragment. The adapters are used to attach the fragments to a chip, with each fragment attached to a different place on the chip. Because it would be impossible to see the signal from a single strand of DNA using the Sanger sequencing approach, each fragment is amplified by a process similar to PCR to generate enough DNA to sequence. Sequencing of each fragment on the array occurs one base at a time. All of the bases to be added are labeled with a fluorophore, and each fragment is extended by one nucleotide. The chip is then imaged with a high-resolution camera, allowing the first base of each fragment to be identified by its color. Then the fluorescence signal is eliminated and the second base is added. Another round of imaging identifies the second base, and then the third, etc., etc. Because each molecule is fixed in place on the array, sequential imaging allows each base to be successively identified. At the end of the process, a computational output of the sequences of each of the millions of fragments is achieved. In the example shown, the top left molecule has the sequence AGT, while the bottom fragment has the sequence CCT. High-throughput sequencing made it possible to sequence genomes quickly and cheaply, allowing access to more genomes, including those from people and other organisms with genetic traits. There are a number of different high-throughput sequencing platforms, and each varies in some respects. The one most commonly used is the Illumina platform, so let's go through how it works. First, the genomic DNA is randomly fragmented, usually by sonication, and adapters are added to the ends. The sequences of the adapters are complementary to little stubs of DNA on the array surface, allowing each fragment to attach to the array by hybridization. To amplify each molecule to a level that will support sequencing, a process of bridge amplification takes place. This is a series of DNA polymerase-mediated synthesis steps that duplicates the DNA at each step. Once the process is completed, there is a cluster of identical strands around each location where a single fragment initially bound. And since these are all derived by duplication of the initial fragment, they all have the same sequence. And there are enough of them so that when they're sequenced, the fluorescent signal will be strong enough for the camera to see. In the first chemical cycle, each strand serves as a template and is extended from a sequencing primer by one base. This process is the same as in Sanger sequencing, except that only a single base can be added. The added base is complementary to the first base after the primer and is fluorescently labeled, so the camera can read the base added at each location once a laser is applied that elicits the fluorescence. Fluorescence of the first base is then eliminated, and the second cycle adds the next base. The second and subsequent cycles then define the sequence of each fragment, one base at a time. Again, because the location is fixed, imaging at each step can be used to define the entire sequence, usually in the range of 50 to several hundred bases. The resulting short sequences are then aligned with the reference sequence and with each other to assemble the sequence of the organism. Note that the human diploid sequence is about 6 billion bases long. If each sequence is about 100 base pairs, a non-redundant and non-overlapping sequence would require the sequences of 60 million fragments. To be sure the sequence is right, and to capture all of the differences between the two alleles, a depth of sequencing of at least 10 and up to 100 would be needed, requiring sequence of up to 6 billion fragments. Why would we want to sequence a genome? First, the sequence would help reveal the biology of a species, identifying all of the genes and the regulatory sequences that control them. Second, variation from the wild-type sequence could be used to identify the basis of specific traits, defining their biology and the genes involved in the process that led to the trait. Genomes have also been used to trace evolution, comparing the genomes of different species to understand how similar they are and when they might have had a common ancestor, among other questions. For many organisms, but especially for humans, genome sequences have been used to study the history of populations. The studies established that modern humans all originated in Africa, for example, and we all carry a few percent Neanderthal DNA, demonstrating that interbreeding between these hominid subspecies occurred. Genome sequencing has also been used in biological samples to reveal what organisms are present and their abundances. This has been done for the gut microbiome, for example, as well as in samples of water from regions of the ocean. Understanding sequence differences among people can provide insight into their relatedness and origins. And of course, when there is a genetic disease, genome sequencing is an effective way to determine the molecular basis of the disease and develop rational treatment strategies. In the context of human genetic diseases, it has been shown that about 85% of the mutations causing single gene or Mendelian disorders are in exons. This observation immediately suggested a shortcut to determining the genetic basis of all such disorders: sequence only the exons. There are about 250,000 exons that together code for the 20,000 or so human proteins. Since these exons only comprise about 1 to 2% of the genome, sequencing only the exons would be faster and cheaper. But the exons are distributed in short sequences throughout the genome, so how could they be isolated and the other 98% of the genome discarded? Since the human genome sequence and other data identified most of the exons, we know their sequences, and this information was used to design synthetic DNA fragments to capture them by hybridization. First, as for genome sequencing, the DNA is sheared into small pieces and adapters added to the ends. PCR amplification is used to generate enough DNA for sequence analysis. The DNA is then hybridized with a large pool of oligonucleotides corresponding to the exon sequences. The oligos also have biotin. The biotin binds to streptavidin that has been stuck to the surface of magnetic beads. A magnet is then used to fish out the biotin-containing DNA, leaving the non-exon sequences behind. This population of DNA molecules contains mostly exons, and they can be sequenced on a high-throughput sequencer using the methods we learned about earlier. As before, the resulting sequences are aligned to the reference genome, and enough sequences of each base are needed to ensure that the sequence is correct, and that both alleles in a diploid organism have been determined, accounting for variants, polymorphisms, or mutations that might be present. This image illustrates the process. Shown is an idealized drawing of a genomic segment with 3 exons. Below each exon, the targeted region is shown, and there is a series of overlapping capture probes corresponding to the oligonucleotides hybridized to the fragmented genomic DNA. The DNA sequences derived from the captured DNA are illustrated above. There is a series of overlapping reads that mostly correspond to the exons. At each position, it is possible to read down to see how many times each nucleotide is represented. That number is the coverage or read depth for each base, and for exome sequencing should be somewhere between 30 and 100. Again, to be sure that both alleles have been captured and there is enough representation to be sure that the sequence is correct. Here, the exome sequencing workflow is summarized. First, the process of exon capture, sequencing, and alignment that we just went through is shown. After that, a series of computational steps is used to fine-tune the data, after which variants are called. Variants are all sequences that differ from the reference sequence. These may be polymorphisms, which we generally think of as benign or harmless variations, as well as potential pathogenic mutations, which may be responsible for a trait. So how many variants are typically found in an exome? Just the non-synonymous changes, the variants that lead to a protein sequence change, amount to about 10,000 variants per exome. Of these, if you are searching for the genetic basis of a single gene autosomal dominant disorder, only one of these is likely to be the change you are looking for. The challenge is how you find it, and you use your best genetic knowledge to figure it out. First, the causal variant should fit the genetic model. For a recessive disorder, there should be homozygosity for a mutation or compound heterozygosity for 2 mutations in the same gene. In a dominant model, the causal variant should be present in the heterozygous state. If there is a family, the variant should segregate appropriately in families. For recessive disorders, the parents should each carry 1 mutation, unaffected siblings should be wild type or carriers, and affected siblings or other affected family members should have the same mutation or mutations. Dominant mutations should be inherited from the affected parent and shared by affected siblings or other relatives. When the parents are unaffected, the causal mutation should be a new mutation not present in either parent. If you are studying multiple unrelated affected individuals with the same disorder, some or all of them should have mutations in the same gene, depending on whether the trait can be produced by mutations in only one gene or more than one gene. In general, the causal variant should be a missense or nonsense mutation predicted to eliminate or damage the encoded protein. These disorders are generally rare, so the variant should be absent or, for recessive disorders, very rare in the databases of known variants from unaffected individuals. And finally, if the variant changes an evolutionarily conserved amino acid present at the same position in the equivalent protein in other species, it is more likely to be pathogenic. Of these, the most important criterion is that multiple genetically independent affected individuals should have mutations in the same gene, as this would not be expected for random people in the population. This principle was illustrated in the first study using exome sequencing to identify the molecular basis of a phenotype. The disorder is Freeman-Sheldon syndrome or whistling face syndrome. The phenotype has very characteristic facial features and abnormalities of the spine, scoliosis, and the joints. It is an autosomal dominant disorder that results from mutations in a contractile muscle protein, myosin H3. So the investigators were looking for variants present in the heterozygous state. Shown in the figure, are the data for 4 independent cases of the phenotype. If we look at the first row, first column, the first patient has over 4,000 genes with non-synonymous variants, or small insertion deletions, or variants predicted to affect splicing of the introns. Remarkably, successively adding additional cases only cut the number of candidate genes in half. Because the phenotype is dominant and the patients don't generally reproduce, requiring that the variant not appear in the database of known variants, the database was dbSNP in this case, as illustrated in the second row, was a better way to filter the variants. The index patient had 513 genes with variants meeting this criterion, and adding the other patients reduced the number to 53 genes. Going to the bottom row of the table, if the variant was predicted to damage the protein, the index case had 160 genes with variants meeting the criterion, and including the other 3 cases left only the MYH3 gene as the candidate, and that was the mutant gene for this phenotype. As a result of the ease of high-throughput sequencing and computational methods to identify the mutations, there has been a dramatic shift in how geneticists study Mendelian traits. A similar revolution has taken place in studying mutants in other species. Here is a practice problem showing output of sequences from a high-throughput sequencer. See if you can figure out how many sequences there are, what the specific sequences are, and what the sequences of the translated proteins are. The sequences only differ at base number 5, and the data show that there are 2 sequences present. The top 2 sequences are the same and read ATGGGC, and the bottom 2 sequences read ATGGCC. Assuming the first base defines the reading frame, the dipeptide sequences are MET-GLY and MET-ALA respectively. Now apply these data to the pedigree, using the first sequence as the reference and the other sequence as the causal sequence. First, it is important to recognize that this is an autosomal dominant phenotype, as there are affected individuals in generations 2 and 3. There appears to be 50% risk to the offspring of the affected individual in generation 2, and there is a male-to-male transmission. Since the grandparents are unaffected, it is likely that the pathogenic change was a new mutation in their son. If we distinguish the alleles by the one base that is different, all of the unaffected individuals should be GG, while the 3 affected individuals should be GC.